So I want to be the first to welcome you. My name is Brian Striniti, and I'm the Outreach and Engagement Manager with the Dutchess Land Conservancy. We all want to thank you so much for joining us on our first webinar in this year's Earth Matters series. It was so much fun last year to host these events, and we're looking forward to this year's wonderful presenters and topics. Tonight, we're very excited for our program, Jeff Lowenfels on the new soil food web. But first, I'm just going to go over some basic information about the webinar, followed by a brief introduction to the Dutchess Land Conservancy, this program, and tonight's presenter. So first, we'll get the logistics out of the way. You are muted to ensure that there's no interruption during the presentation. Second, the raise hand function is disabled this evening. Please enter all your questions into the Q&A box at any point during the presentation. You can access this box at the bottom center-ish of your screen. You can enter questions there. The questions will be addressed when the presentation is completed or by our team during the presentation if possible. If there are a great deal of questions, we'll try to combine them based on topics as well. Third, chat is disabled for this event for similar reasons. And fourth, closed caption is enabled for this event. To turn on this function, please select the CC icon located at the bottom right of your screen. And finally, I would like to hand the mic over to Julie Hart, the Dutchess Land Conservancy's Education Director, who will tell you a bit about our organization, this webinar series, and our guest presenter tonight. Julie.
Thank you, Brian. Good evening, and thank you all for joining us tonight. I'm Julie Hart, an ecologist and educator here at the Dutchess Land Conservancy. Many of you are probably familiar with the DLC, but for anyone who's not, let me just give you a quick overview. The Dutchess Land Conservancy is a private nonprofit land conservation organization located in the Hudson Valley of New York State, and we're dedicated to preserving the farms, forests, wetlands and waterways, open spaces and wildlife habitats of Dutchess County. We are an accredited land trust, and since our founding in 1985, we have worked with hundreds of landowners and protected almost 46,000 acres of land. And many thanks to the Molly B. Schaefer Education Fund for sponsoring this event tonight. We're so excited to be kicking off a new season of Earth Matters, the DLC's winter webinar series. We'll be bringing you interesting speakers on the first Tuesday of each month through April. And this season, we are digging into the topic of soil health. Warning, there may be some puns ahead. We'll be learning about soil food webs, regenerative soil management practices, composting, and the ecology of soil health. Our theme is very simple. Soil is not dirt and should not be treated like dirt. So let's get started. Tonight, we're thrilled to welcome Jeff Lowenfels as our speaker. Jeff is the author of the groundbreaking, I did warn you about the puns, groundbreaking series of books, Teaming with Microbes, Teaming with Fungi, Teaming with Nutrients, and the just released Teaming with Bacteria. He's also a lawyer who writes the longest running garden column in North America and never having missed a week in 41 years. The combination of garden writing and law has earned him the moniker of America's dirtiest lawyer. Jeff is the former president of the Garden Writers of America, a GWA fellow, and in 2005 was inducted into the Garden Writers of America Hall of Fame, the highest honor a garden writer can achieve. Through his writing, Jeff has become a leader in the organic gardening sustainability movement, and we're excited to have him as the lead-off speaker for our series this year. So with that, I'll turn it over to you, Jeff, and Jeff is going to talk about the new soil food web. New science will make you a better and organic gardener.
Well, thank you very much. That was a great introduction. I probably shouldn't have put this slide up as the first slide. Probably about 15 or 20 people hung up as a result of seeing it. But what I want to talk to you today involves these two words, rhizophagy cycle and let's see what's going on here with my, there we go. But it's really all about the new soil food web. Just again, to add on to the introduction, I used to call myself the America's dirtiest lawyer. I had 2 books out then I had a trilogy. So that's when I started calling myself Lord of the Roots. And now I've got a 4th book out called Teaming with Bacteria. So I've got a quadrilogy of books. Let's get down to work. Let's get down to business. This is an Alaskan sled dog doing his business. What I want to talk to you today is about something that is brand new. In fact, so new that I would say out of all the people in New York State, you 50 to 100 people are probably the only ones who are going to learn about rhizophagy. And it's something that we were talking about a little bit earlier. Next year at this time, when you have a seminar series, this will be given and accepted and extremely well known. But I'm happy to be the individual who gets to introduce this to you. It's called the New Soil Food Web Discovery, and the discovery is called the rhizophagy cycle. But before we get into that, I want to talk to you about the soil food web. The soil food web is nothing more than a series of food chains that occur in the soil. The little guy gets eaten by the bigger guy, gets eaten by the bigger guy, and there's thousands of these chains in the soil. Well, every now and then, something on one of the chains looks up and sees something on one of the other chains and eats it. And so it connects the two chains. And the next thing you know, you get this incredible web of linked chains indicating who eats whom in the soil. That's the basis of the soil food web. And I could give the whole talk on this one slide. The way the soil food web operates, just as a summary, is that the photosynthetic energy of the plant is used to make exudates. About 50% of that energy is used to make things that drip out of the root system, exudates. These are high carbon molecules, and they're right there in the rhizosphere, right there in the root system, right up against the roots. They attract bacteria and fungus that need the carbon that are in these particular exudates. You're exudating right now, by the way, you're sweating, and your sweat is attracting bacteria and fungus to your skin. In any case, in the soil, those bacteria and fungi are happily living next to the roots, and along come the things that eat them. Protozoa, which you studied in high school, nematodes are something you didn't study in high school. Protozoa are paramecium, amoebas. The nematodes, which also come along, are true worms, very microscopic worms, and they eat bacteria and fungi, and they do so because they also need the carbon. Well, this carbon is useful, but they don't need all of the nutrients that are in the bacteria and the fungi, and so that excess is, for want of a better word, pooped out into the soil. And it happens to be in plant usable form, because in order to get into the plant, it has to have a charge on it. That's described in a book I wrote called Teaming with Nutrients, and you can learn about how the nutrients get into the plant under this system and what they do once they get in there in Teaming with Nutrients. But what happens is they're deposited in the soil as a result of all of the eating by the nematodes and the protozoa of the bacteria and the fungi. So there are actually three ways plants get nutrients. The first way is what I just described. This is the system that we discovered, or that Dr. Elaine Ingham popularized and discovered around 2006. Teaming with Microbes came out and sort of popularized the idea. And the scientific name for it is microbe-mediated solubilization of soil nutrients. So you get the poop is excess nutrients are in the soil, and they migrate through the soil to the root system. Then we added something called the mycorrhizal fungal symbiosis, because what we were discovering is that in addition to the system described above, the microbe-mediated system, that there were fungi specialized that were going into the plant and trading for these exudates and giving the plant water, a lot of the metal nutrients that it needs. A tremendous amount of nutrients came through this symbiotic relationship of mycorrhizal fungi. That was around 2014. And in fact, it was so important that I wrote a book on it called Teaming with Fungi to explain that system. Well, we thought that was all. And who needs any more for the plants to feed themselves? But then we discovered the role of endophytic bacterial symbiosis. Endophytic just means in the plant, inside the plant. And there are certain bacteria that literally spend part of their life inside the plant, helping the plant and getting something from the plant. It's a symbiotic relationship. So this is what I really want to concentrate on today so that you can understand the full picture. But in order to do so, let's take a look at the 2006 soil food web and see how it was described. And it's really very, very simple. It all happens in the soil. And what happens is the nutrients are released and they go into the plant. This is a generalized depiction. The exudates are many. Plants are capable of making a tremendous number of molecules. In fact, everything the plant needs is made by the plant inside the plant cells. And this is a listing of some of these exudates. Now, this is what they look like when they're dripped out of the tip. This is what it looked like in 2006. This is what it looks like today with more modern photography. And you can see these are where the exudates are released. And this is the bacteria and the fungi coming in order to take advantage of these exudates. These are the root hairs. This is a single root. So let's get a really better depiction of how this worked. I'm using big words. Let's make this simple. We've got a bunch of plants that are sitting around and it's a hot Friday. And one of them says, I'd like some Japanese food. And one of them says, no, let's make it Mexican food. And another one says, no, I want French food. Well, it turns out that the Japanese choice wins the vote. And so the root system drips out special. This is the root system. Let me turn it over so you can pretend these are the roots. It drips out a specialized exudate designed to attract Japanese bacteria. And that's what it does. And if it wanted American food, it could change the exudate and it would get American style bacteria and fungi. The plant is in control. And these wonderful nutrients move in through the soil to the cell because the microbes put a charge on them. This is the cell, and this is the series of different ways that each individual nutrient, and there's a different way for each one to get into the plant so that the plant can make all those exudates and everything else it needs. All right, so the plant is in control. It can make the exudates that it needs so that it can get the nutrients necessary to make the part of the plant, for example, that is growing. The plant is totally in charge. Not you, the gardener, which of course upsets people quite a bit. Let's see here. Got to get this. Again, there are these three ways they get the nutrients. I've just described this mediated soil. Let's take a look at the mycorrhizal fungi system, which is part of the soil food web. The fungi are capable of going out into the soil. They're much smaller, obviously, than the main root. They're capable of getting into pore spaces, and they bring back a tremendous number of things, phosphorus in particular, nitrogen, water. You can read as well as I can. All of these things are brought back and fed into the plant. This is how it operates. You've got two seedlings sitting out in the sun. One of them says, I'd like some bologna. And lo and behold, it makes the exudate necessary in order to attract bologna fungi. It puts it out into the soil. And the next thing you know, it attracts the bologna fungi. And the bologna fungi feed the plant the bologna. What's so great about these mycorrhizal fungi is that they stay and continue to feed the plant if you don't disturb them. And the plant gets bigger as a result of these mycorrhizal fungi feeding the plant. And even more important, they share their goodies with other plants near them. And so you get this mycorrhizal fungi worldwide web that's become so popular and that people read about. It is a very major addition to the 2006 soil food web. And so now you've got the nutrients moving through the soil, and now you've got nutrients moving through fungi feeding the plants. And now we've got this new thing, this endophytic bacterial symbiosis, which is called rhizophagy. This is something that was discovered and in 2010, perfected a couple of years later, and it involves bacteria. And the thing is, we really don't know much about bacteria as gardeners, even though they're incredibly important. The bacteria come in different shapes. They produce different substances. This is where our antibiotics come from. These are really, really important, and they're everywhere. So plentiful, I think, that we take them for granted. So this is two pictures. These are tips of needles, a needle and a pin. And these are the bacteria coating the needle and the pin. Unbelievable. They're everywhere. They multiply every 20 minutes. And the 5-second rule is just silly, because the minute you look at something, you get bacteria on it. This happens to be a single cell inside a tongue. And these are all the bacteria associated just with that single cell. They're very, very pretty, some of them. They have unbelievable abilities to move. They've created these flagella that are literally propellers. And at microscopic, electron microscopic level, you can see that they actually look like outboard motors. They're absolutely phenomenal. As noted, they breed every 20 minutes. And man, they are really something. These are some of the flagella that enable them to move. They're so small that they can't move in liquid like you and I can. If they had oars, they would just simply stay in place. And so they've developed these propellers that spin around. And again, they're everywhere. And when you think about them, you shouldn't be thinking about a bacterium, which is a single bacteria. You should be thinking about bacteria, tons and tons, gigantic numbers of them. There's really no such thing as a bacterium. So think bacteria. And one of the things about bacteria that are in the soil is they're capable of producing phytohormones. These are hormones that help plants grow. Gibberellin, auxin, cytokines, ethylene, they're unbelievable and helpful. The plant can produce them, but the bacteria can do so as well. And so when they're endophytic inside the plant and producing these things, they are helping the plant germinate, grow, produce flowers, fruit, go into dormancy, drop their leaves. All of this activity, it turns out, is heavily influenced by bacteria. Who knew? Very few people. So these are endophytic bacteria. We really should be somewhat familiar with this, because nitrogen fixing rhizobia, which people use on legumes, are endophytic during part of their life. They form around a root hair. They cause the root hair to form a nodule, and inside the nodule, they're deprived of oxygen, which makes them capable of producing nitrogen, which is what they do. And so again, these endophytic bacteria are really, really incredible. You get plant growth promotion, you get all sorts of terrific things, and they get into the plant in the root system. When the root system branches, they enter in through the cracks, they get into the plant, they move through the xylem phloem, they can get in also through stomata, and they can get in through various injuries that the plant might have. And there they are in the plant, producing these phytohormones and doing wonderful things for the plant, and even more wonderful than we know. Here are some that are entering into the stomata. These are the bacteria, and they're getting into the stomata. And once they're in the leaf surface, they do some unbelievable things, including fixing nitrogen, which is what rhizophagy is. So let's talk about rhizophagy. This is the thing that I mentioned at the beginning, that really you are the only people in New York State probably that know anything about it. It's just now coming to the public fore. I pronounce the word rhizophagy. Rhizo meaning root, phagy to feed on, root feeding. And the woman who discovered and named this particular process involving endophytic bacteria has a name I can't pronounce. She is Australian, and she discovered in 2010 that bacteria were inside the very young meristem root cells at the tip of the root. Right down here, she began to see these cells with bacteria in them. Nobody had ever seen them before. Again, we know that they are attracted by the exudates, and the meristem cells are all down here. And through the kind of photography microscopes that she's able to use, she's able to see the bacteria themselves in a way that we really never saw them before. And what she theorized when she saw that these bacteria were inside the plant cells was that the plant was literally opening up the cells, letting the bacteria in. In other words, eating, like in Little Shop of Horrors, these bacteria inside the plant. Well, her funding must have run out because that's where she left it. Incidentally, I pronounce it rhizophagy. For a couple of years, the professor, Dr. James White at Rutgers University, who took this information and then expanded upon it. We've been going back and forth about how to pronounce this word because the Australians pronounce it rhizophagy, and that's their Australian accent. I say it rhizophagy, that's my American accent. So that's how we say it. And here is a picture again of the root tip. These are cells that are sloughed off. And this is where the microbes were found to enter into the plant. And then up here are the root hairs. So we've got this distance here. It's a very interesting process. So let's go back to 2006. You remember those bacteria that are attracted by the exudates. They're eaten by the protozoa and the nematodes, but some of them are a little stubborn and they smell something that causes them to act a little differently than the other bacteria that are in the soil. They smell a popcorn, a buttered popcorn smell, butyric acid, and they think that that is signaling them to leave the bacterial slime colony that they're living in and move to a new bacterial slime colony that doesn't have so much competition. But in fact, that smell tricks them. It's being produced by the plant, and it tricks them to break through the cell wall of the meristem cells, the very thin ones at the tip of the plant. And so they break through the cell wall and they enter a little space that's known as the periplasmic space. The best way I can describe this is to show you a tofu package and have you imagine that white outside plastic part, that's the meristem cell wall. It's very thin, it's very immature, and the bacteria go through that cell wall and find themselves in that water-filled space around the tofu itself. The tofu would represent the cell insides where the cytoplasm is with all the organelles, and it has a membrane around it. And then there's that water space, that's the periplasmic space. And lo and behold, the bacteria find themselves in that periplasmic space, and here's a picture of them in that space. And we know that when they go into the plant, a couple of things happen. Here's another picture so that you can see them in bigger numbers. They're inside the cell. They're not inside this part. There are some in there, but they're mostly in this periplasmic space. And they're circulated around this periplasmic space. And as they circulate around, they're multiplying. They're producing ethylene, which causes the cell to stretch and grow. And they're producing nitrate because they're being sprayed by the plant itself, the cell itself, to try to kill it. because bacteria are not really supposed to be inside plant cells. And so that spray that the cell produces is called ROS. It's an oxygenated spray, and it strips off the cell walls of the bacteria. So it's this oxygenated spray, and really what it is the plant trying to defend itself from the bacteria that are now inside that periplasmic space. And it's called ROS, or reactive oxygen series. When you get a sunburned, that's what's causing it, ROS. And I even found a sunburned ROS to represent what's going on here. But in any case, the bacteria had their cell wall stripped off by this spray of oxygenated. And in order to weaken that spray, the bacteria produce ethylene, which causes a little bit of dilution and the cell to stretch. And they also produce this carbonic acid through a bunch of chemical reactions that weakens the ROS. It's an antioxidant, so that the bacteria continues to live without its cell wall as it circulates around the plant. Now, the plant is spraying this ROS. It's got to make sure that it doesn't destroy its own cell wall, and so it strengthens itself during the process. So the cell wall is being strengthened. It's being stretched by these wall-less bacteria that are circulating inside the plant, and they're generating nitrite, which gets converted into nitrates. Nitrates are then absorbed along with the cell walls into the plant cell, where they feed the plant. So you've got this little trap system going on. The microbes, if they stop producing the ethylene, they don't get fed some of the exudate by the plants. And if the microbes stop producing nitric acid, they're going to be completely destroyed. So there they are. They're producing nitrogen. They're fixing nitrogen inside the cell. Up to 40% of the nitrogen that a plant uses, who knew, is produced internally by this fixation system. And they're producing these cell walls, which are getting eaten, and they contain lots of the micronutrients that plants need. Who knew? I mean, this is just sort of a crazy, crazy thing. And all the while, they're being circulated around the outside of the cell by a circulation current. And so they're being circulated around, and they're producing and feeding the plant as they circulate around. Now, at some point in time, because they form these quads, by the way, some of them are sixes, and that's what they begin to look like as they begin to multiply, and you begin to get more and more of them. And at some point, you get so many of them that they get caught up against the meristem cell wall. And instead of they're being circulated, some of them get caught in the cell wall. Instead of producing the ethylene and having it be circulated, the ethylene gets concentrated. And the concentrated ethylene causes the plant cell to form a tube. And that tube is something you and I know as a root hair. It's a tube coming out of the meristem cell. It's not a separate cell itself. It's a tube. And it's caused by these bacteria producing ethylene. Plant hairs are not there or not formed to produce or to take in nutrients. They're there because the bacteria create them. And they're slowly, so there's a tube from the main, and here are all these hairs. These are all being produced by bacteria producing ethylene and pushing out of meristem cell. And each one of these is attached to or a part of a meristem cell that's filled with the bacteria. And what happens is the bacteria begin to fill these tubes, and lo and behold, the tubes get filled with the bacteria. And when the bacteria become too plentiful inside the tube, they're producing ethylene, remember, they're ejected out of the tip of the root hair. And this can happen 3, 4, 5 times as the root hair is growing. And this is a picture of the bacteria. And these blue spots are bacteria piles that have been created. They're put back into the soil where they grow back their cell wall. And in a few days, they go back into the plant again and repeat the rhizophagy cycle. Here is a picture of the bacteria inside the cell hair. And they're going to be blown out of the tip where they'll grow back. See, they're producing a marker which can be identified. They're thrown out into the soil. And again, many times, one root hair, and they regrow their cell walls. Now, I don't want to give you the impression that these root hairs don't absorb nutrients, because they do, but they do it later on, as after the explosions have taken place. They're still very thin cell walls, and so they absorb the nutrients. And again, if you want to know how this all happens, teaming with nutrients explains how that happens. So they're back in the soil again. And really what we've got in this rhizophagy system is a ranching system, a sheep system. So the bacteria are taken into the plant, they're attracted through the cell wall, and they're shorn, and a couple of the lamb chops are eaten, and then they're put back out to pasture again. Whereas the original soil food web was simply a farming system where the plant exudates attracted the populations, they grew, and they put the fruits, so to speak, out into the soil, and they were gathered and taken in. So what's really happening is you've got plants that are both farming and ranching at the same time. And it's really a terrific thing. And the fact that these bacteria, some of them are eaten and pooped out, and some of them are rhizophagy cycle, and they repeat the cycle, is really an amazing fact. And this is a depiction of the rhizophagy cycle. They go in, they circulate, they increase, they produce the root hairs, they exit, and it just continues around like crazy. So plants really do eat bacteria, and it's a very important thing, because up to 30% or 40% of the nitrogen in the plant is bacteria. How important is it? Every single plant that has root hairs has the bacteria rhizophagy cycle going on. All plants with root hairs, and you've never heard of it, and now you know about it. It's really a stunning new discovery that really adds on. And there's a gentleman named Dr. James White at Rutgers University, who with his students seems to be the gentleman leading the new discoveries, and there's more and more coming, which we will describe. Now, a lot of your plants have trichomes. Tomato plants has a trichome, cannabis plants have a trichome, and it turns out that these trichomes, when I wrote the book, we thought it takes a long time for a book to be published. So I wrote the book 3 years ago. And when I wrote it, we were theorizing that these trichomes, they look like root hairs. They're probably formed the same way, but nobody could really confirm it or deny it. And the discovery has been made post-publication. And what has come to fore is that inside these trichomes are bacteria, and they're taking some carbon from the plant cell, and they're producing nitrogen. The carbon that's being fed to them is plentiful enough so that you don't get excess oxygen. You need an oxygen-free environment to get the nitrogen fixation. So it's a beautiful little factory inside all of these trichomes, influencing your cannabis and influencing your tomato plants. Again, there are endophytic bacteria all over the place. And there's some of the rhizophagy, they're definitely endophytic, but the other ones get in through these little cracks that open up. They go in through the system and they travel throughout the plant, again, producing these wonderful phytohormones. And some of them, a good representation of them, get up into the area where a flower is formed, and they get trapped inside the seed when it's formed. And these bacteria live inside those seeds until the seed germinates. And when the seed germinates, the bacteria that are inside the seed and in the seed coat themselves, they jump into the soil so that they can feed the root, rhizophagy, and act as the 2006 version of the soil food web. And they move into the plant and up into the plant. And so it's very important never to sterilize your seeds. If you're starting seeds in a paper towel, you need to plant the paper towel along with the germinated plant in order to get the maximum amount of bacteria. It's really a very, very important aspect. And again, if you want your plant to have access to all of these wonderful hormones, you want to definitely make sure that you're not sterilizing the seed, that you're not sterilizing soil. You want these things. These are known as plant growth promotion hormones, and they help the plant deal with both abiotic stresses, heat, cold, rain, that kind of wind, as well as biotic stresses, all the pathogens and attacks that the plant gets. Because when the bacteria enter into the plant, they strengthen the plant because the plant has to strengthen itself in order to be able to handle the bacteria. And so, for example, here's a picture. It happens to be in, I think this is in Houston. If you notice, a part of the area doesn't have snow. That's because those plants are infected with an endophytic bacteria that changes the freezing point of water. Some of these bacteria produce things that drip out of the plant, like cytophores that can cleate minerals. They can tie them up so that they don't poison the plant. They tie them up in the soil, or they can bring a particular metal into the plant that the plant might particularly need. So these are really very, very important. And there's some really neat ones that you can already buy. These are things that are already on the market, and more endophytic bacteria are going to be introduced to the market, because these are the things that the big, what I used to call evil chemical companies, are beginning to study. They're studying the biology, because now they know that you can have the right kind of endophytic bacteria invade the cells of a wheat plant, for example, and feed it 30 or 40% of its nitrogen. They're hoping to be able to feed it 50, 60, 70% of its nitrogen. That means you don't need to use the chemical fertilizers, which really are not good for our environment and not helping the world. So you've got these wonderful things. These are used throughout the world. These fix nitrogen. This is one that many cannabis growers are beginning to use. Fixes nitrogen. This one takes out some of the bad fungi that get in the way of your tomato plants. This particular one, these particular ones form spores, so the shelf life is easy, and these have to be freeze dried. These are pink pigmented facultative, which brings up the fact that a lot of these bacteria are hard to pronounce. We're going to have to learn how to do so. So there's some very obvious takeaways that should come from the soil food web as it's now described. You don't sterilize seeds. You don't toss your soil unless you have a root problem with the plant that you grew in it. You reuse it because it's full of the exudates that are needed to attract the right bacteria and fungi. It's full of the right kind of bacteria if you plant the same plant in that kind of soil. So for example, if you plant your tomatoes in a container, there's no reason why you shouldn't be planting your tomato in a container every single year, the same soil. It should be getting richer and richer if you add the organic matter that you're taking out with it. So obviously another thing is you need to buy Teaming with Microbes, nonetheless Teaming with Bacteria, so that you understand what the soil food web is. Now there are some not so obvious takeaways that present some pretty interesting opportunities. It turns out that the bacterial mix in heated compost versus the bacterial mix in vermicompost, compost made with worms, is different. And some plants do better with some bacteria than other plants. And so now that we know that the bacterial mix is different, and now that we know that rhizophagy bacteria are different depending on what kind of plant you use, or endophytic bacteria are different depending on what kind of plant you're growing, then it makes sense for people to experiment. Your particular crop may do better with one kind of compost than the other. And so this is a great opportunity. When you make compost, by the way, you probably don't, oh, gee, I keep doing this. I think I can go backwards. When you make compost, consider making compost from the plants that you're going to be using the compost on, to try to get the maximum benefit of the right kinds of bacteria, because the bacteria is different depending on the plant. So for example, when you have one kind of a tomato plant versus another kind of tomato plant, the soil itself will basically have the same bacteria, one tomato versus another. But the endophytic bacteria that are inside each tomato plant will be different and specific to that kind or variety of tomato plant. This is what makes heirloom tomatoes. You've got your right mix of bacteria for your particular area. Too much of a good thing is a bad thing. So chemicals, we know, have an impact on endophytic bacteria. We know they have an impact on soil bacteria. When you use artificial nitrogen, i.e. fertilizers, chemical fertilizers, they reduce the microbial impact. They reduce something known as the nitrogen efficiency. And so these are not good. By the way, when you use chemicals and you don't end up with the same amount of bacteria, you are losing soil structure, because the slime that the bacteria make in order to stick to each other, in order to hide from the enemies that they have, is what begins the production of soil structure. The bacterially slime aggregates get tied together by fungi. They're not flat. This is where air can come in, where little guys can hide from the bigger guys, where nutrients can be accumulated. This is the beginning of soil structure bacteria. And so if you're using chemicals, you're not completing the system the way it needs to be, and you don't have the bacteria that you need to have. We don't use pesticides. We let the soil food web take the system out. It's all so important that we not mess up soil food web. And we tend to, as gardeners, because we think we're in charge instead of the plants. We're not treating soil bacteria properly when we do massive farming. We are told that we only have 60 years of soil left, 60 harvests left. That's not very many. It's a scary thought. And it's really because we are not protecting and using the soil food web the way we should. It's really pretty obvious. The plants should be left in control. There's a way to do that for almost for every plant that you do grow. They're amazing. They just do all sorts of phenomenal things. And a lot of the phenomenal things they do as a result of the bacteria that they take in. Now, I always tell people, if you're not convinced, just think about this. Oh, what happened? Not convinced, think about this. And then, of course, I have my book. Think about the redwoods. Whoever fertilizes them, whoever sprays them, they team with the microbes in the soil food web to get to where they are. There's no reason why every single plant that you have can't do the same thing. So basically now, folks, you know a lot about the rhizophagy cycle and endophytic bacteria. You now have the new soil food web. So let me see what we can do. I think do I stop the share? Is that what you want me to do, Julie, Brian?
You can leave this slide open with your books on. I think that's
okay.
Good advertising, right?
Sure, exactly. Now, I always tell people, you buy this one, and if you don't think it's helpful and the best book you've ever read on soil, I'll buy it back at 3 times the price. So this is the one you want to start with, folks. Then I would go to teaming with bacteria, even though it's the fourth one written. And then I would go with teaming with fungi. These are how plants get their food. This is what the plants do once they have it. All fascinating stories as far as I'm concerned. It's something that every gardener really needs to know. So.
That's awesome. Thank you very much, Jeff. We very much appreciate your conversation here tonight. And we'll be sending out an email when we'll include links to your books and maybe even put that good order in that you mentioned, because with a quadrilogy, it could get a little confusing. So that's awesome. Thank you so much.
It could be, and I hope nobody took any notes because it's all in the books, and obviously a lot more than I was able to touch in. I see we've got some questions. You want me to answer the ones we've got?
Sure. Yeah, I just wanted to remind folks before we do that to keep an eye on their email for the links from tonight's presentation, and also to sign up for our subsequent Earth Matters webinars, including next month, Dawn Smith, The Answer is Soil, on Tuesday, December 6th at 6 p.m. But yeah, now let's just, looks like we've got some great questions coming in, and feel free to keep asking them. So I'll give the first question to you, Jeff, here, if you don't mind. So in terms of plant evolution, when did rhizophagy appear? And does this occur with lichens, mosses, and fungi?
Well, again, I always tell people I didn't invent this stuff. I just summarized what other people and dumbed it down so that I could understand what other people do. So I hope I'm answering these questions the right way. Rhizophagy appeared when root hairs first formed. I can imagine the mycorrhizal fungi. Fungi were one of the things that enabled plants to live in terrestrial situations. That it was way back at the beginning. The other part of the question was, do lichens, mosses, or fungi show rhizophagy? No, they don't. Now, that doesn't mean that there aren't bacteria on all of them or even in them, but the rhizophagy cycle is something that's plant-based, as far as I know.
And the answer is always going to be changing as research moves along, I'm sure.
Yeah, it probably is.
Now, the next question is, under this approach, can you still add biochar to your soil?
Absolutely. This doesn't change the way we use the soil food web. This just adds other ways we can make it better. So biochar is as people know, a great condominium for microbes, including bacteria. And so as long as you charge the biochar, it's a phenomenal addition to your soils. I'm a big proponent of biochar. Now, there are problems people have with biochar. Is it efficient to make it? And the prior, is all those questions. But as far as using it, it's great. Does it hurt the rhizophagy cycle? I would imagine it helps it.
Great, very cool. Good to hear that about biochar. So next question coming in is, what are the implications for all this on crop rotation? For example, don't plant nightshade family plants in the same spot yearly.
Yeah, I think we're going to have to, great question. We're going to have to study a lot of these things. It's very interesting that this stuff just began in 2010. We don't know the answers to a lot of these things. So would I continue to change, for example, potatoes? You're not supposed to grow in the same place 2 years in a row because of scab. Yeah, I would continue to rotate those. Would I continue to rotate tomato plants and pumpkins and things like that? I don't think I would. I think I'd do the experiments to see whether it works, but I would try to plant them in the same areas.
Okay.
I guess to refine that answer, if it's got something to do with disease or it's got something to do with pH, then go ahead and rotate. But unless it does, why?
Okay, there's a similar question that mentions the planting tomatoes that you had spoken about before. In this case, they're asking planting peas and beans in the same spot each year. Is that something that would be a good idea or a bad idea?
Yeah, why not? I think it's a good idea because it's got the nitrogen from all of those nodules. When the plant dies, the nitrogen from those nodules goes into the soil. So yeah, it ought to be a great idea, great place to grow. And you might not even need to add the rhizobia. In other words, roll your seeds in the rhizobia because it's probably already in the soil.
Very cool. Next question coming in, I love because it's about my level of gardening. So how do houseplants manage to grow for years on sterilized potting soil?
Well, I guess the real answer is it's not sterilized. Usually the question is, does rhizophagy work with houseplants? Of course it does, because they have anything with root hairs. By the way, if you take the bacteria out of the plant, the root hairs don't form. The roots begin to grow up to the surface. They get all messed up, and then you put them back, they grow normally again. Whoa, it's amazing. So the answer is the soil's not sterile. Simple as that. It's got organic material in it. Let's start there. The plants, as you know, are producing exudates, even though they're in a pot, and those exudates are attracting goodies, and those goodies are feeding the plant. Otherwise, the plant would die.
All right, and now we have a question for an average gardener. What steps would you take to take care of the soil during the beginning of the growing season versus the middle and end of the growing season?
Sure. Well, since we're at the end of the growing season, I would make sure that I put down all of my organics right now. All the organic foods that feed the microbes. If you put it on the soil right when you're planting, it may not be available. It takes a while for the microbes to break it down. So you give it the winter in order to do so. You mulch on top of it with the appropriate kind of mulch. If it's an annual garden, straw is a terrific mulch. Grass clippings, as long as it's not got a lot of seeds on, it's terrific mulch. If it's a perennial bed, your leaves are a terrific mulch. And if you grind the leaves up, it's a great mulch for your vegetable garden as well as the green straw, et cetera, et cetera. So you want to put that stuff down. You want to mulch with organics right now so that the organics are worked into the soil by the worms, et cetera, et cetera. And even though we get frozen soil, we end up with a little teeny layer between the soil and the snow cover. And there's tremendous amounts of microbial activity. I'm told more so, although it's hard for me to believe, more so in the winter months than during the summer months, but you get great breakdown. All right, so then springtime comes along, do not rototill. All of your plants the first year of this system should be rolled in mycorrhizal fungi, which you can purchase at good nurseries. If your nursery doesn't have mycorrhizal fungi for sale, it's not a good nursery. You roll your seeds, you roll any transplants in mycorrhizal fungi. You put it into the garden without rototilling the garden. If you need to make a row, you take a 2x4 or a tomato stake and you just make the row. You don't rototill, which breaks up the soil food web. It breaks up the mycorrhizal fungi. It puts the bacteria that are supposed to be in the root zone in a completely different location. When you cut a worm in half, you don't get two worms. You get two halves of a dead worm. So you plant carefully, but you use rhizobia if you're talking about legumes. Let's see, what else do you do? You provide support and you label everything. And then you put the mulch around the plants that the plants need. If they're annuals, we're talking green mulches. If they're perennials, we're talking brown mulches. And you step back and you let the plants do their thing. And then you harvest them. You take one of your rows and you bring it to someplace that needs it. Feed the hungry, plant a row for the hungry, and you take the rest and you enjoy it yourself. Did I get through the whole year? I think I did.
The whole year in under 2 minutes. Good job, Jeff.
Yeah, there you go. There you go. Yeah, I think the most important thing is to put the foods down now. Now, in fact, it's funny. In Alaska, the big the big controversy for a little while, I wrote a column and I said, do not rake your leaves. Leave them on the lawn. Leave them. And this was a month. We're earlier than you are, obviously. This was a month or so ago, and I get a couple of wise guys who go, "No way, you're smothering a little lawn," all this. No, I don't tell my people to do stuff if it doesn't make sense, and if I haven't tried it myself. But in any case, today there are probably 10 what I call clickbait articles on the internet about don't rake your leaves. All of a sudden this year, that's become the hot trend. And the reason it's a hot trend is because of the soil food web that we've been talking about. And all of these microbes will break down these leaves and make your yard a better yard next spring. Oh, I forgot, you got to put in your spring flowering bulbs right now as well. I like to use a little bit of mycorrhizal fungi on those, and I like to put a little mulch over them as well.
I personally like the no rake approach because it speaks to my lifestyle of not wanting to rake leaves.
Well, that's why we call it teaming, T-E-A-M. Let them do the work. We spend too much time working. That's not what we want to be doing. Yeah.
I agree. All right, well, we have a couple of questions here. There seems to be some tilling information needed, additional information needed. So someone is asking if they have a small garden that's 20 by 12. They've asked if they should rototill or not, and they've been told it's okay because it's a small size. And then someone else was saying, are you saying it's okay to take a no-till approach to your soil preparation? So to till or not to till?
Don't till, period. And I love to till more than anybody. My father had one of the biggest rototillers you'd ever see. Oh God, it was so much fun to use. I have 2 or 3 of them sitting around in my tool shed right now, but they are really bad. The reason we rototill, it's funny that it comes up in this situation when we've been talking about rhizophagy, is because Jethro Tull, not the band, but there was really a guy named Jethro Tull. He was an English lawyer, and he had a big estate. And back then, people believed in what was known as the humus theory, that roots literally ate soil. Well, now we have the rhizophagy fact that roots eat bacteria, but back then they thought roots ate soil. And so Jethro Tull came up with the idea that, gee, if that's so, then we should pulverize our soil. And so rototilling was invented, and who adopted it? Well, Thomas Jefferson, John Adams. These people were all growing here in the United States. And when you break up this mycorrhizal fungal network, what you're left with is a bacterially dominated soil, which is what row crops like. And so using a bacterially dominated soil in America made a lot of sense. We were taking old growth forests where it was pretty hard to grow a tomato, and we were converting it into these open fields, blah, blah, blah. So yeah, rototilling is a no, big no. And double digging is a big no. And French digging is a big no. We've all seen seedlings coming up through the sidewalk. People complain about plants growing through their tarmac in their driveways. The plant doesn't need you to rototill. Nobody rototills in nature.
Wow, I think I speak for all of us when I say it's really nice to know that the things that take the longest and the most amount of work are the things you should not be doing.
Yeah, right, exactly.
So we've got a quick question here. Could you repeat the order in which people should be reading your books?
Yes, start out with Teaming with Microbes, and if you don't like it, I'll buy it back at 3 times the price. Don't tell any homeless people, otherwise I'm going to end up with orders of 500 cases. Teaming with Microbes, then Teaming with Bacteria. So you get a full picture of the soil food web. You add Teaming with Fungi, and then Teaming with Nutrients.
Thanks, Jeff. And we'll send that order out in the email subsequent to this presentation, because I'm going to remember that myself, because I gotta start digging into this stuff. I'm going to go down to the question at the bottom, because for some reason it keeps getting pushed back. How do you make compost for every different plant?
Well, it's very difficult, and I guess the answer is you don't. But if you're a tomato grower, if that's your big hobby, then it makes sense maybe to have a separate pile where you take all the old tomato parts and you make compost out of that. Or it may turn out that you do have plants that do better with vermicompost than thermal compost. So you want to have two piles of a vermicompost situation and a thermal compost situation. You know, if you're retired like I am, you know, you've got a lot of acreage and tremendous amounts of time. You know, you might be able to make 7 or 8 different kinds of compost, but you know, it's pretty hard to do.
Thank you. The next question is about tillage again, but this is a little bit of a another approach to it. If you plant a cover crop, how can you not till it under in the spring? Or do you do some form of minimal tillage in that case?
Yeah, you can do minimal, obviously, or you can crimp it. In other words, you break the plant off, or you take a weed eater and cut it down to the ground. That you don't need a rototill. That's a big no-no. Big no-no. You know, you think about that 60 years left of soil because it's blowing away. Why does it blow away? Because it doesn't have structure. Why doesn't it have structure? Because they're using combines and rototillers.
Yeah, that makes a lot of sense, actually, thinking about the large amount of agricultural land and the large machines that hulk through them.
Yeah.
This is a good question and kind of relevant to what I would like to do with my yard a bit. So if you don't till, how would you start bed, vegetable or flowers from scratch if you don't want raised beds? So I think just cutting into
Sure.
your lawn or whatever it might be.
Right. Well, you know that may be an instance where you rototill the one time. But on the other hand, you may also decide to put a garden in by bringing in soil and putting it on 6 or 7 sheets of newspaper. Everything underneath it'll die off, and then you've got your garden there with the soil that you know is good, maybe your own compost. But if there's ever an exception to the rototill situation, that's it when you start the garden. Although I got to say, if you've ever rototilled the lawn, you get little pieces of the root and little pieces of the dandelions, and boy, they're in your garden for a long time. So it makes more sense really to lay down some paper or cardboard and then put soil on top of it.
And now we have not so much a question as a comment from someone who tried no mow May last spring and says it got out of hand and they wound up with amber waves of grain. And his wife thought he was shirking his mowing duty. So I can see how some of these could cause a little bit of discord in families.
Yeah, you know, it's interesting. I wrote this article I think I mentioned about don't rake your leaves, and I got a wonderful letter which was in my incidentally, if anybody ever wants to take a look at my columns, they come out every Thursday at www.adn.com, Anchorage Daily News, which is the newspaper that's in this new TV show called the Alaska Daily, if you've seen it. They don't have the garden columnist in the background, I can tell you that. But I wrote the article and I got a letter from a 10-year-old girl. And she said, gee, that was a great article. And yum, 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 worms love leaves. But I make my money raking leaves. Could you tell people in Anchorage that if a 10-year-old knocks on their door, to let her rake their leaves? Which was pretty funny. But I had to write back to her and I wrote a column and I said, look, you know, every now and then there are things that we don't like. You know, we got to take care of Mother Nature first, even though it's not so good for us. So here's some other things you can do instead. But when it comes to using the soil food web, however, there is no other choice. This is the science of how plants grow. And so when people start using chemicals and use the chemical system that we all used before 2006, when the soil food web became, known, we are not doing a good thing for us, for the environment, for our kids, and it's hard to understand why, as gardeners, we don't understand that, but we need to. And if we don't, our kids won't be gardening.
Hear, hear. All right, I think we are out of questions, which is a good thing. It means you were very thorough and answered. You got 15 questions answered and about 10, 15 minutes. So great job, Jeff.
Yeah. If anybody has a question they want to ask, I don't get to my email every single day, but they can write to jeff@gardener.com and I'll try to get to the answer. So now, if I could, I want to make clear to everybody that I did not invent any of this stuff. Dr. Elaine Ingham is the guru of the soil food web. Dr. James White came up with a tremendous amount of research with regard to the rhizophagy. Both Dr. Elaine Ingham and Dr. White publish papers every now and then. And so use the gardener's best tool, Google, and get an alert so that when something comes out on rhizophagy, when something comes out on Dr. Elaine Ingham and Soil Food Web, when something comes out on Dr. James White, a new paper, you get it, and you will continue to be among the only 200 people that are advanced and up to date in Dutchess County.
The myophagy elitists, or the rhizophagy elitists here.
Right, right.
All right, well, we've got a couple other comments coming, and just thank you very much for a wonderful presentation, and people learned a lot and really enjoyed your fun slides and very entertaining delivery.
Thank you. Thank you. Appreciate it. All right, well, let's do this again when we get another discovery.
I'm sure there's one right around the corner. There's
Right.
always something new in the soil science world.
We'll all be looking for it, I'm sure.
Yep.
Yes, sir.
Well, thank you so much, Jeff. We really appreciate your time. I think we've all learned a lot, and we're all thinking about what we're going to be doing next year in our gardening plans and how we're going to make our soils more healthy.
Great. Terrific.
Right.
Me too.
All right. Thank you, Jeff. And thank you for all of our attendees tonight. What a great time, and have a good evening.
Thanks so much. Good night, everybody.
Good night.
Good night. Good night.